is Dr. Konstantinos Mitsakakis from Freiburg. He studied physics and biotechnology, and he is doing fascinating work in towards new diagnostic tools. And I look forward to your presentation, Multi-Pathogen Identification on a Centrifugal Microfluidic Platform. This is a new project. Please. You're still waiting for... Okay.
So thank you very much, Reto, for the introduction, and I think that your presentation couldn't be a better introduction to this presentation, which in this presentation I'm representing the Framework 7, Framework Program 7 funded project called Diagnostic, standing for disc-shaped point-of-care platform for infectious disease diagnosis. I think after your presentation it is a bit needless to say what is the need to emphasize what is the need to have point-of-care tests for infectious diseases, but just to emphasize a little bit from the report of the WHO from 2013, despite the efforts, there are still recorded 207 million cases of malaria and about 630,000 deaths from the same disease per year. Most of them are focused on the resource-limited settings you see here in the map, mostly around the tropical zone. And when we say resource-limited, it is in terms of infrastructure, training, and affordability. Now, an additional problem in these regions is that people who present themselves with acute fever, they may not have malaria, but there might be other diseases with the same clinical symptom, and this makes the diagnosis even more difficult. And this, in turn, leads to a significant percentage of false diagnosis. It is registered even up to 30%, and this, in turn, leads to wrong drugs and wrong treatment. So the aim of the project that we started about a year and a half ago there are two specific objectives. First of all, to achieve a differential diagnosis of acute fever, meaning which pathogen is responsible for the fever that the patient presents, and second, to use this diagnosis in order to properly guide the healthcare provider for the suitable treatment. That means to provide antibiotic or antimalarial or some other kind of treatment. The diagnostic panel that we chose for this project was, as you see here, malaria, dengue, typhoid, and pneumonia. The reason why we chose these diseases is primarily because they are four of the major killers, if I may say it like this, and also because from the clinical point of view, they represent three main pathogen categories, parasite, virus, and bacterium. And we want to demonstrate on our platform that we're able to identify all the three of them with one run. Then there's another problem which rises, for example, for dengue. There's often the case that the viral load, so if the patient presents himself within the first 5-6 days, the disease of dengue can be diagnosed through the viral load or through the antigen, but then if he presents himself later, then these indicators are over, and you need to have others like IgM in order to detect the disease. So this we took also into consideration for our panel. And therefore, you see here that we are trying to achieve a bimodal detection, which means through both nucleic acid and protein detection. It's quite challenging because nucleic acid means DNA, but also RNA, and protein means antigens, but also antibodies. For the nucleic acid detection, we use a LAMP technology, which is an isothermal amplification. It's suitable for both types of nucleic acids. It offers high specificity because it uses a set of 6 primers per target, and it's typically faster than PCR. And for the immunoassays, we use antigens and IgM antibodies, as I mentioned earlier. So what happens is that our partners from the project who are responsible for the assay development, they prepare the protocol on a benchtop in tube, and then they deliver the protocol to us. And what we do is we translate these tasks into what we call unit operations. For example, preamplification, valving, mixing, aliquoting, everything that is done on the bench. We try to simulate it in a workflow, and then this workflow is translated into microfluidic structures. And each structure is a modular entity, and we can interface those structures in order to finally have what we call the lab disk platform, which is basically a CD-shaped cartridge, and it uses centrifugal forces, so we spin it like a CD, centrifugal forces for liquid handling. And there's a major advantage in that it needs no external pumps or tubes, and this in turn leads to a high degree of automation, portability, and point-of-care use. Well, the CD footprint, as I said, it allows high parallelization and multiplexity. And, well, typically, like a CD needs a CD player, we have the lab disk player, which is the disk processing device comprising of several units. For example, for disk rotation protocols, acceleration, deceleration, and so on. A thermal unit, which is capable for user-defined temperature protocols for isothermal or for PCR amplification. Integrated magnets for magnetic bead handling, and optical unit for fluorescent or luminescence detection. The aim is to have what we call a sample-to-answer analysis. This means that we are aiming in the end to have blood uptaken from the patient, inserted into the disk, and have the result at the end with minimum external intervention. What I said earlier about the modularity is visible here, so I have highlighted with colors different regions, and each region is serving a particular function. For example, the purple is for sample inlet, whole blood, that is. And the green area is for sample preparation. This means that what could be done typically on the bench, like to lyse the bacteria or the pathogens, extract DNA, purify, and so on, it is done on this green area. And then the gray area is where you mix it with the amplification reaction mix. And finally, this yellow place, this yellow area, is for aliquoting. I would call it the analogous to a multi-pipette. And then in these chambers in the perimeter, you have the amplification because you have pre-stored dry primers. And we managed to achieve high automation because in these blue regions, we have stored all the buffers that are needed for this washing, purification, and so on. And these buffers are stored in these aluminum pouches. And then by centrifugation, they are released. The liquid is released. Then the ultimate goal is to have what I call all-in-one, which means nucleic acid analysis and immunoassay parallel running to the disk. It's not utopic because the recent results from a month ago design data show that this is pretty much feasible. And here I would like to show you two videos. One has to do with the fabrication of this system. It all starts with this milling machine, which we provide a computer design, and the milling machine is with a micro driller, is drilling in a plexiglass, the design. And then we use this design as a mold in order to fabricate the negative, which is this red disc that you see here, which is PDMS. And then the PDMS, we insert it in a hot embossing machine, and then we place the foil, thin foil, above this. It is heated, and then by applying air pressure from the top, the foil is taking the shape of the mold. And then we have the disc ready. And then when we have the disk ready, we have to develop the proper protocols, as I mentioned earlier, temperature, frequency, and so on, in order to have all these functions on the disk, like lysis, conjugation of the DNA, of the extracted DNA with magnetic beads, washing of the magnetic beads, purification, and so on. So I would like to show you a few short sequences of these unit operations. So here, excuse me. So the first step would be a centrifugal release of the mixture of the sample together with the magnetic beads in this chamber. Cell lysis takes place, and then there is a magnet that will collect the beads in a pellet. You'll see it like here. And a second magnet will take the pellet and bring it to the second microfluidic chamber, and with centrifugation it is released. And so we are, these steps are continuing, and then we reach this area where we have this multi-pipetting step that I told you earlier. So the sample goes there, and then by increasing centrifugation, it is distributed to reaction chambers. Here we increase the centrifugation, and then with software, embedded software, we have the temperature protocol for PCR or for isothermal amplification. Right. So I mentioned about the fabrication. So the technology is called microthermoforming. It is typically used currently for pill blister packages and pill production. And we have adapted to the microscale in order to tackle the challenges of the microfeatures. We use, as I mentioned earlier, foils, polymer foils, which is much advantageous because it is more compatible to large-scale production. And here you see schematically what this large scale means. It means that you have several stations serially, one next to the other. The first one is doing the structuring, the second one is doing this pick and place of those aluminum pouches that I told you earlier, and the other one is doing the sealing, cutting, packaging, and so on. So instead of a conclusion, I would like to summarize what are the main technological innovations of this project and what impact they lead to. Okay, so there was some problem with the presentation. So basically, the technological innovations are, first of all, that we have developed this universal buffer, which is capable of lysing parasites, bacteria, and viruses. The second innovation is that we have this scalable production technology, which means that we can achieve volumes of the order of a few tens to hundreds of thousands in the near future, 1 or 2 years. And the other technological innovation, very important in my opinion, is what we call the openness, the open nature of the platform, meaning that it consists of modular components, and then based on the geographic needs in Africa, for example, or in other places, and based on the diseases which are more prevalent, we can change the biochemistry by keeping the design stable. So once we complete this design by the end of the project, it would not be difficult to target it to particular locations that are in need based on the prevalence of the diseases. So as future steps is the clinical validation, which is to take place in 2015 through contacts of 2 partners of ours, Institut Pasteur in Senegal through our partner University of Sterling, and in Nyankunde Medical Center in Congo through the partner University Hospital Basel and the group of Patrick Hutzinger. Post-project technical steps would be the alternative power supply, for example, battery or solar panel operated, would be to ensure long shelf life of components at high temperatures, to achieve standalone use, no laptop, and to upscale the disposable production technology. And some non-technical future steps would be to resolve regulatory issues, cope with cost, and pursue follow-up and parallel funding. So as I mentioned in the beginning, it's a European Commission funded project, and we acknowledge for their funding. The project ends in end of 2015, and I want to thank very much all the partners that are participating with the excellent expertise that they contribute. And Clinam, for example, is one of the partners, and as I mentioned earlier, Patrick Hutzinger with his group at the University Hospital. I invite you to visit us on the website, and I thank you very much for your attention.
Thank you very much, Dr. Mitsakakis. We have time for 2 or 3 questions. Please.
I wasn't clear, was this 1 disc per patient?
Yeah, correct. Yes, that's right.
And roughly how long does the analysis take for, you gave an example earlier, previous speech.
Yeah.
Had 15 minutes for a result, so how quick would you expect to see a readout?
Right. So from the moment that you insert the blood from finger prick until the analysis, we would estimate between 60 and 80 minutes at the moment, but we still have much time to progress with that. And progress means reduce the sample preparation time and run parallel the immunoassay with the sample preparation time. That would, I would expect that would reduce at least 20 minutes.
What's the ideal time?
The ideal time? Well, I guess it depends also on the type of the test. So if it is a rapid diagnostic test, it's 15 minutes, but then it is targeting something totally different. It is targeting one marker. It is targeting only immunoanalysis. So considering the fact that we are targeting also the nucleic acid, we have to extend our time somehow. I believe that 40 minutes would be pretty okay. Considering also, of course, how many patients arrive in a medical center per day, I know that it is much desirable to reduce the time as much as possible.
Very impressive.
Yeah. Thank you.
Yes, I wanted to ask you, how do you compare the results of your, with what do you compare them? I mean, do you have some controls or calibration method in your...
That's a good question, thank you. So part of the project, so our partners are finishing the development of the bioassays, and immediately the next step, before we go to clinical validation to the African sites, will be to compare with the so-called gold standards, which means malaria, we should compare our results with the DISC, with the rapid diagnostic tests, and optical microscopy possibly, but certainly PCR. And the other diseases, the bacterial diseases, for example, also with PCR, in order to compare, as you mentioned, the sensitivities between the gold standard and our approach, so that we know when we go to Africa what we should expect.
Okay,
and what are the limits.
In the DISC, you don't have a calibration.
So there is the negative controls and positive controls, if that's what you mean.
Yeah.
They will be included, yeah, of course.
Okay. And another thing, how much volume do you use?
Right.
Because for a fingerprint, you can take just 25 microliters or less, maybe.
No, we have, we investigate a concept that it can give us up to 100 microliters, and this is how much we use with fingerprint. Yeah.
With one disc, can you test for all the diseases at the same time, or only, let's say, for one disease? Because, as you said before, fever is something which is usually happening in most of the diseases. So, is it possible to test for all the diseases at the same time, or you already have to have an idea what maybe it is, and you get a yes and no answer?
The ones that we have, the panel we have on the disk, they will be analyzed at the same time, if this is what you mean. If we need to increase the number of the probed diseases, it is possible. There is space on the disk. Then there are other issues coming, like the cost of the reagents that you need to have pre-stored. So it's different if you have reagents for 5 panels or for 10 or for 15. And that's why I think that, that's my personal opinion, a low number of diseases tailored to the needs of some particular areas would be maybe more commercially viable. That's my personal opinion.
Yeah, congratulations, that's a great system. I was wondering, what do you predict that the cost will be for the machine and for the discs?
Right. Yeah, it's typically a question that we are asked. So it definitely depends on the volume, on the production volume. So I can say about the discs that are fabricated in our group. So a colleague of mine from the business development, they have made a scale-up costs, and if the volume goes up to a few of the order of 100,000s, then the cost for the manufacturing can be down to a few euros for the plastic disc. Then we have the additional cost of the reagents from our partners, but as far as I know, that is of the order of €0.50 per per reagent per disease. For the reader, at current we are using expensive components because we need to have the proof of principle. But optimizing these components, so the camera and fluorescent unit and so on, it could be down to a few thousands of euros.
I think we have to stop here.
Okay, thank you very much.